All right, I guess we should start. It's always a pleasure to have Alec Kimmelman. He was, and it's a particularly real fun thing for me to have him back. And as he reminded me, both times I've hosted him, we never got to have dinner because last time he was a faculty here. So Alec did his, well, he's right now the chair of radiation oncology at NYU and a professor there. But right before that, and this wasn't started in 2016, Alec was here in our department in radiation oncology and was assistant and associate professor here. So we started our labs at the same time on the same floor. So it's really fun. He did his MD PhD at Mount Sinai and then came to Boston and to our department for his residency. So I can go through the list of things Alec has accomplished. I will not do that. It's a little, and because that will come with stories. So I'll just tell you the one piece of his work that I understand is the first paper he published, because that's the time when we used to actually read each other's manuscripts. But this was a seminal discovery, the Genes and Development paper. That was the first paper from his lab, showing that pancreatic cancer is dependent on autophagy. And right after that, he went on to publish more on this topic of, really understanding how autophagy contributes to pancreatic cancer therapy resistance, which was, I think, a Nature paper in 2020, and more of that. On another side, which is all, again, metabolism and pancreatic cancer related, he discovered a new pathway where, in regard to iron transport with a factor NCOA4, with Joe Mancius was the first author on that, faculty here now, and Alec and Joe discovered this new pathway, which he has continued to publish a lot more on. So I will stop there and let Alec do the talking. It's really a big pleasure and pride to have you back here.
Thank you, Dipanjan. Thank you for the invitation. Thanks for the nice introduction. Is my screen showing correctly here?
Yep, it's fine.
Thank you. Well, it's good to be virtually back where I spent the largest chunk of my career. Would have been nice to see old friends in person, but Zoom will do. These are my disclosures. And I'm going to be talking about some off-label use of a couple of compounds. So my lab studies pancreas cancer. Most people in the audience are likely very familiar with it, but it's really one of the worst cancers that we see. And it really can be summed up by this one statistic about a 10% 5-year overall survival. And unfortunately, this has been a substantial improvement, actually. The philosophy that we've had in my lab for the past many years has been that perhaps some of this intrinsic therapeutic resistance points toward altered metabolic pathways. I'm not going to talk really anything about genetics today, but the defining genetic feature of pancreas cancers is nearly all of them have activating mutations in the KRAS oncogene. And our group and others, and many others, have shown that it's critical for tumor development and tumor maintenance in pancreatic cancer. However, given the biochemical nature of the protein, it's been thought to be an undruggable target. However, revitalized interest over the past several years has now led to a resurgence in targeting RAS, in particular, direct inhibitors to the protein, such as G12C, are now showing promise in clinical trials. Although rare in pancreas, it's much more common in lung. Other inhibitors are making their way down the pipeline. So we look forward to that. All right, we're not chemists in my lab, so we basically try and spend our time on this side of the diagram here, which is we try and understand how RAS and pancreas cancer generally may rewire their metabolism and how that could be targeted. So I'm going to talk to you today about examples of 3 themes that we study in the lab. Many are interrelated, but we sort of artificially distinguish them. One is we study central carbon metabolism and fuel source utilization in these tumors, and we've done a lot over the years with glucose and glutamine. As Dipanjan mentioned, I will sort of summarize our long journey of studying autophagy in pancreatic cancer. both cell autonomous and non-cell autonomous work that started when Dipanjan and I first started our labs together, and then also some work that we've done in terms of the role of the tumor microenvironment and nutrient sharing networks. So really, to summarize a lot of our metabolic work over the past decade or so is that these tumors have incredible mechanisms of metabolic plasticity. And this is an example of a pathway we identified a number of years ago that utilizes glutamine through a series of biochemical reactions that lead to increased NADPH that allows the cells to maintain redox homeostasis. And we were excited because the Calithera developed an orally bioavailable clinical grade inhibitor to the first enzyme of this pathway, glutaminase, which allows the carbon skeleton of glutamine to be used in the form of glutamate. And what we quickly learned from this work was that within a matter of hours, the entire metabolome of the cell and the proteome governing the expression of these metabolic enzymes was totally rewired, and that led to alterations in metabolic fluxes. And what that actually meant was that after several days of strong growth suppression, all of a sudden the cells just started regrowing again with the same kinetics as they did before. In addition to their ability to metabolically rewire, they're also professional metabolic scavengers. So work from Cosimo Camiso, Daphne Barsaghi, and others have shown that pancreatic cancers can take up extracellular protein and use it to fuel their metabolism. Work from our lab and others has focused on their ability to degrade themselves through autophagy to help fuel metabolism. And then we and others have identified these interesting, or at least I think they're interesting, metabolic sharing networks where other cells in the tumor microenvironment can actually share nutrients with these cells. So they have an incredible way of scavenging nutrients when nutrients are poor. So this led us to really want to try and understand a way to study metabolism or metabolic dependencies in vivo. And this is a collaboration with my good friend and faculty member, Dana-Farber and LeBrode, Andy Aguirre, and by a very talented, just graduated PhD student, Doug Bianco. So we set out to use functional genomics to try and identify globally metabolic dependencies in pancreas cancer, but also compare those that are in vivo to those that are in vitro, because there's been a lot of discussion in the field about how well 2D cultures in nutrient-rich media model metabolism in a tumor. So we felt doing this functionally might give us an insight into that. So we used a cell line derived from the B6 background KPC model, which is a KRAS-driven pancreatic cancer model. And then Doug created a custom CRISPR library that really covered all of the metabolic genes in the genome and a whole bunch of control genes. And then we did a variety of dropout screens in vitro, in 2D, in vitro, in 3D, and then in vivo in both a syngeneic model, BLAC6, and also nude mice, so we can kind of understand what the role of the immune system or the adaptive immune system was playing in metabolic dependencies. And one of the striking findings from this study was that there was an excellent correlation between the metabolic dependencies in 2D on a plate to those in vivo, much higher than that we actually expected. And put another way, 86.5% of all the hits were seen in vivo, but there was an additional 13.5% that you would miss in vitro. In contrast, while most of the hits were seen in vivo that were also in vitro, there's almost 40% of those that are likely spurious and might be due to in vitro artifact conditions. So you'll see pretty much the overwhelming majority of hits in vitro, but things may be overrepresented that are not truly in vivo metabolic dependencies. Similarly, on a pathway level, a lot of the canonical metabolic pathways that we and others have been studying were very similar both in vitro and in vivo. Things like the TCA cycle, nucleotide metabolism, glycolysis, et cetera. Another aspect of the screen that we did, and I mentioned, is that we wanted to see if 3D culture might better model metabolic dependencies than 2D culture. So Doug performed a similar screen using a 3D assay and basically grown in Natrigel. And similarly, there was a pretty high correlation between 2D and 3D metabolic dependencies. However, when you put all the screens together, we saw some interesting patterns in certain genes. One of them is squalene synthase, or FDFD1. And this is basically a key component of the mevalonate pathway and the first committed step of sterol biosynthesis. And when you look across all of the different screens that we did, FDFD1 was not a hit in 2D in vitro screens. It showed some dependency when we moved to 3D screens, increased dependency in vivo and nude mice, and the greatest dependency was seen in the syngeneic system. So this was a very interesting hit to us because it was one of those that would be missed in vitro, and there also may be an immune component given the stronger phenotype seen in the B6 mouse. So we knocked out this gene using CRISPR, and you could see confirming the screen data that there's really no impact on growth in 2-dimensional conditions. However, in 3-dimensional conditions, you get a large suppression of growth. You can inject these orthotopically into the pancreas, and you can see compared to control, clones, or pools, that FTFD1 knockout cells fail to form robust tumors in the pancreas of these mice. And you can rescue this tumor growth by re-expressing a cDNA in the CRISPR cells. And also consistent with the pattern we were seeing where the phenotype was the most robust in the B6 mice, you saw a significant influx of CD8 positive T cells into the tumors where FTFD1 or squalene synthase was deleted. You can just see that quantitated here. The other thing that was very exciting to us about this target potentially was that there's actually a pharmaceutical or clinical compound that is a very potent inhibitor of FDFD1. It's TAC475. It had been designed originally as a cholesterol lowering medication because it would block the mevalonate pathway, but it was suspended in randomized clinical trials due to some low-grade liver toxicity, which would obviously be unacceptable in a healthy population for just cholesterol lowering, but would probably be acceptable in an oncological population given the grade of the toxicities. So we wanted to see if we can recapitulate the genetic data by treating mice with TAC475. And even as a monotherapy, we were able to slow the growth of pancreatic cancers implanted in B6 hosts. And similarly, to what we saw with genetics, you got a nice increase in CD8 positive T cells in these tumors. So to summarize this work, the majority of the metabolic dependencies that we see in tumors are recapitulated in 2D culture. The in vitro metabolism probably overestimates the contribution of some metabolic pathways. And this, we think, provides rationale that a lot of people are already doing to refine some of the media formulations that we use to better recapitulate the in vivo situation. I didn't show you this data, but certain metabolic dependencies are masked in these screens by metabolic crosstalk or nutrient sharing. The 3D culture may better model certain metabolic dependencies, such as those that affect cell signaling. And we think that FTFD1 is a promising potential therapeutic target due to cell intrinsic effects as well as immune-mediated effects. So I'm going to change gears here and talk about some of our work in the tumor microenvironment and how that impacts pancreatic cancer metabolism and growth. And this is work done by a very talented postdoc, Robert Bond, who has just started his own lab here at NYU. So, you know, one of the defining features of pancreas cancer is its desmoplastic reaction. When you look at these tumors under the microscope, you'll see scant tumor cells, but there's a tremendous amount of fibrosis and extracellular matrix that's deposited. It's a really complex mixture of many cell types, often the minority of which are epithelial tumor cells. It's poorly perfused and likely nutrient poor, and it's one of the most hypoxic tumors that we see. So we've been working for some time on how different components of the tumor microenvironment may share nutrients with the tumor cells. And when Robert came to the lab, he became interested in looking at neurons in this regard. And the reason for that is pancreas cancer is one of the most highly innervative tumors that we see. For the clinicians in the audience, they'll know that many of these patients actually require interventional procedures like nerve blocks. The pain is so bad. And a lot of groups have been working on how the nerves might be contributing to tumor progression. And really seminal work from Tim Wong's group has shown that, sensory neurons and sympathetic neurons are pro-tumorigenic in pancreas cancers, and cholinergic neurons are tumor suppressive. And really most of the focus has been so far on how the nerves may secrete neurotrophic growth factors, and that may provide growth stimuli to the tumor cells. But Robert speculated or asked the question, could these peripheral axons from these neurons metabolically support pancreas tumors under nutrient deprived conditions? And if you think about it, nerves are the perfect metabolic conduit to do this, because the nerve body is in the ganglia, which is well perfused and nutrient rich circulation, and the axons travel all the way down into the tumor. So it'd be the perfect conduit or funnel to take nutrients from the nutrient rich circulation and bring them into the nutrient poor tumor. So to do this effectively, Robert utilized a device that separates, where you grow the neurons in one side, the axons grow through these little grooves, and hydrostatic pressure separates these two different compartments, because you really don't want to study what's being secreted by the nerve bodies. You want to look at what's being secreted by the axons. So Robert utilized these systems and rat dorsal root ganglion neurons to see at first what metabolically neurons were secreting. And interestingly, neurons were secreting actually a fair amount of metabolites out of their axon in this side of the chamber. One of the ones that, or two of the ones that jumped out of us were serine and glycine. And the reason for that was there's been an explosion in interest in serine as an amino acid in cancer over the past several years. Work by a number of labs have shown it to be important, and in a large part because it does a lot of things that tumor cells need. It's important for phospholipid synthesis, 1-carbon metabolism, redox homeostasis, to make nucleotides, glycolysis, et cetera. So we wanted to understand what was going on with serine and what role the neurons play. And this is just to highlight that most serine in the cell, when it's synthesized, is made from glucose. Interestingly, when we looked across human pancreatic cancer cell lines, there are plenty, as has been shown by Karen Bowsden's lab, that are serine independent, meaning that when you take away serine from them, they're perfectly capable of growth. But a significant minority of those, about 40%, actually were serine, or we call them exogenous serine or ex-serine dependent, meaning when you remove serine from them, they can no longer proliferate. So we went across a whole panel of cell lines. About 40% require the presence of serine in order to proliferate. And the reason for that, at least biochemically, was quite straightforward. These tumor cell lines don't express PHDDH, which is one of the rate-limiting enzymes of serine biosynthesis when you're making it from glucose. Many also don't express PSAT, which is another critical rate-limiting enzyme. So, and we did a bunch of tracing experiments that, not surprisingly, these cells that don't express these rate-limiting enzymes, they cannot make glucose-derived serine. Now, what was very exciting to us was that when we looked across a large panel of human pancreatic cancer specimens, that almost the exact same percentage of these lacked PHDDH, indicating that what we're seeing in culture is replicated, or actually what we see in people is replicated in culture. So to look at this functionally, Robert then turned back to these chambers and he grew cells, pancreatic cancer, serine-dependent pancreatic cancer cells in the wells, either in the presence or absence of neurons. And you can see summarized here in this graph that neurons were able to significantly support the growth of multiple different serine-dependent pancreatic cancer cell lines grown in the absence of serine and glycine. So we next set out to try and understand what serine was doing in these pancreatic cancer cells. I mentioned, it does many things. So we wanted to understand what its key contributions were. And this is going to sort of be unfair to Robert, but through a ton of work, he basically showed that it really wasn't doing any of the canonical metabolic pathways that many people have shown serine plays a role as in cancer. But the one aspect that pancreas cancers couldn't figure their way out around was you need serine to translate RNA into protein, which isn't that surprising either. But what was surprising was that there appeared to be some selectivity in the decreased translation. And to look at that, Robert did a number of things, but he performed a riboseq, which is a technique where you basically look where ribosomes are stalled on the transcripts and determine where the stalling is by next generation sequencing. And what we saw here was fascinating, was that there was actually selectivity to where ribosomes were stalling in the absence of serine and glycine in pancreatic cancers. They were stalling on two different serine codons, TCT and TCC. So you can actually look across the genome and try and predict what proteins would be more and less effectively translated by the relative abundance or the amount of the codons TCC and TCT. These are the ones that were the most sensitive. And so when you look at the proteins or the pathways that are enriched in these different fractions, the top one was secreted soluble growth factors, and strikingly, NGF was one of the top genes that was enriched in that would be most predicted to not be affected by serine and glycine deprivation. And indeed, when you took serine-dependent cell lines and you cultured them in decreasing concentrations of serine, they started to secrete significantly more nerve growth factor. So this was all in vitro work. We wanted to see if this was operative in vivo. So we took a serine-dependent pancreatic cell line, orthotopically implanted into the pancreas of mice, and then put mice on either a normal amino acid diet or a serine glycine-free diet. And you could see that the tumors were significantly smaller in the mice that were on the serine glycine-free diet. This appears to be a decreased proliferation and not an increase in death. We don't see really any change in apoptosis markers, but we see a significant decrease in phosphohistone H3. Now, what was really interesting was that when we looked at the tumors and tried to see how innervated they were, there was a substantial increase in nerve innervation in the tumors that arose in the serine glycine-free diet. And you can see that here by this three-dimensional clear imaging compared to those in the normal amino acid diet. And when you quantify that, you can see there's a significant increase in neuronal innervation in the tumors that were in the mice that were fed the serine glycine-free diet. So the next thing that we wanted to understand is, can we capitalize on this for some therapeutic purpose? And what we did was we made use of LOXO-101 or larotrectinib. This is a TRK inhibitor, which is the nerve growth factor receptor. Oncologists in the audience will be very familiar with this. It's an FDA-approved drug for cancer treatment, but it's actually different from what we wanted to use it. It's approved in tumors that have the TRK translocation, where this now becomes a tumor-specific driver. However, we wanted to use this to actually block TRK signaling in the nerves and basically prevent the innervation or the increased innervation in the low serine glycine conditions. So we did the same exact experiment as we did before, except this time mice were randomized to either get control or LOXO-101. First, we wanted to see if it was having the effect on nerves that we expected. So again, in the serine glycine-free conditions, a lot more nerves grow in these pancreas tumors. When you add LOXO-101, you completely block this. And similarly, although LOXO-101 does very little on its own in normal dietary conditions, serine glycine diet on its own decreases tumor growth. You get a significant improvement in tumor decrease when you add LOXO-101 to the serine glycine-free diet. Similarly, same thing, proliferation follows the same pattern. And again, it was very important for us to understand whether this was something happening in human patients. So we went back and in collaboration with Waman Wang from MD Anderson, it's a very large collection of clinically annotated pancreatic cancer patients, TMAs. We saw a very striking inverse correlation of PHDDH and expression and nerves. So meaning that tumors that had high levels of PHDDH, meaning that they were able to biosynthesize serine, had very low levels of neuronal innervation. Those with very low or absent levels of PHDDH, meaning that they can't biosynthesize serine, had the most nerves seen in those samples. Similarly, you could do the same thing in the TCGA using NGF mRNA, and you could see a very nice inverse correlation in the TCGA. So Robert went on to really explore how the codon bias was working in these samples. I'm happy to talk about that, but I'm not really in questions, or if people want to talk offline, but I'm not going to present that data here. So the model is that Robert came up with was that cells that are serine independent, whether or not they're in serine rich or serine deprived regions of the tumor, grow just fine. They can biosynthesize serine when needed. However, in serine dependent pancreatic cancer cells, they grow fine when the serine is abundant, but in areas of the tumor where serine may be depleted, what happens is the tumor cells, through increased transcription and selective translation, secrete nerve growth factor. This leads to an increased recruitment of nerves into the tumor and provides serine for them to grow. So to summarize this part of the talk, peripheral neurons can secrete amino acids such as serine that can metabolically support pancreas cancer growth and survival in nutrient-deprived regions of the tumor. Although I showed you that pancreatic cancers are professional, they can rewire their metabolism extremely well. mRNA translation and protein synthesis is still a bottleneck for tumor growth. And serine deprivation in the serine-dependent pancreatic cells decreases overall mRNA translation rates. However, they have the highest decreased translation efficiencies against specific codons, namely TCC and TCT. And what this allows is biologically adaptive responses to occur, sort of stopping things like proliferation and allowing transcripts like nerve growth factor to be more effectively translated. So with the time remaining, I want to talk to you about our work in autophagy, in particular because it started at Dana-Farber in the early days when Dipanjan and I started our labs together. And we've been working on it all these years, and it's now actually in the clinic and showing signs of efficacy. So it's something I'm excited to sort of talk about the genesis of how this all started. So for those who are not familiar with the pathway, autophagy or self-eating is a mechanism that cells have to survive under periods of stress. The most well-studied stress is starvation or amino acid deprivation. It's regulated by a number of things, namely the mTOR pathway is one of the key regulators, as is AMPK for energy charge. I'm not going to go through the biochemistry of how the pathway works, but there's a cascade that allows the formation of an autophagosome. In these autophagosomes are the cargo, which are damaged protein, protein aggregates, whole organelles. Autophagy is really the only way a eukaryotic cell has to get rid of a whole organelle like a mitochondria. These become enveloped in the double membrane autophagosome that fuses to the lysosome, where the contents are degraded that can be recycled back into the cytosol for a variety of bioenergetic or anabolic reactions. One of the interesting things is we actually have medicines, while not specific to inhibiting autophagy, through their function on the lysosome, do inhibit this pathway. That's chloroquine, the anti-malarial, and hydroxychloroquine, the anti-rheumatological drug. Chloroquine's been used since the 50s in people. And they work by deacidifying the lysosome, which basically prevents the degradation of the autophagosome. So while not necessarily specific autophagy inhibitors, they act to inhibit the end of this pathway. Now, based on the cargo that can end up in an autophagosome, it's not surprising that autophagy can really fuel nearly every aspect of cellular metabolism or central carbon metabolism in a cell, depending on what cargo is degraded. And work that Dipanjan mentioned, that Joe Mancius, who's now a faculty member at Dana-Farber, did when he was in my lab and Wade Harper's lab, identified the role of autophagy in another type of metabolism, namely iron metabolism. And Joe is doing some fantastic work, continuing to work on this. So more than a decade ago, we made an observation that even in nutrient-rich conditions, pancreatic cancers had very high levels of basal autophagy. And this is just showing how they have fluorescent, this is a fluorescent marker for autophagosomes, where non-transformed cells, or quote unquote normal cells, when they're in nutrient-rich conditions, really have very low levels of autophagy. And we went on in a series of studies to show that these pancreatic cancers do rely in varying degrees on autophagy for proper growth. This is a simple xenograft model treating with chloroquine. You can see you prolong survival in these mice. Annabin Mitra, in a collaboration with us, treated the Hopkins PDX panel with hydroxychloroquine. You can see pretty much across the board, with rare exception, there was responses to hydroxychloroquine as a monotherapy. You can recapitulate this by knocking down various essential autophagy genes. This is ATG5. And then we crossed the KPC, or the genetically engineered mouse model of pancreas cancer, to Noboru Musashima's conditional deletion of ATG5, and you can prolong survival from pancreatic cancer in these mice. So all of this was tumor cell autonomous, or what autophagy was doing to support the growth of the tumor cells themselves. We became interested, as I mentioned, in the tumor microenvironment and its role in this, in particular because of the sort of the defining desmoplasia of these tumors. And Chris Salza, when he was in my lab at the Farber, identified a really interesting autophagy dependent pathway in the stellate cells, which are the type of cancer-associated fibroblasts in these tumors, that allowed them to secrete non-essential amino acids, in particular the alanine, at very high levels. The alanine fueled the TCA cycle, allowed more efficient oxidative phosphorylation to occur, allowed carbon sources to be used for other things. And this allowed the tumor cells to grow in nutrient poor conditions, which is what we believe they're seeing in the tumor. And through yet an unidentified secreted factor, the tumor cells actually can increase autophagy in the fibroblasts, which again provides this feed forward loop, essentially calling for food. And in vivo, what this means is if you take these are human pancreatic cancer cells and you co-inject them with stellate cells, you can increase the tumor take and the speed of tumor growth. However, if you block autophagy in the stellate cell compartment itself by using hairpins, either ATG5 or ATG7 in the stellate cells, you lose this enhanced tumor growth when you do the co-injection studies. And this is a human system, and this is the same thing in the syngeneic mouse system. So we showed that autophagy has critical functions in a cell autonomous fashion in pancreas cancer. In particular, I didn't show you all the data for this, but it seems to support various aspects of metabolism, such as mitochondrial metabolism. This is showing how you decrease oxidative phosphorylation when autophagy is inhibited. That leads to decreased proliferation. And you also have a non-cell autonomous effect when you use systemic autophagy inhibition, in that you block this metabolic crosstalk that I just described, where stromal cells can feed tumor cells metabolites, and together this increases the anti-tumor efficacy of autophagy inhibition. One of the things that we noticed when we were doing some of these experiments, that there was a significant contribution of the immune system in the anti-tumor effects of autophagy. And this is something we want to explore further, and this is a collaboration with Rushika Pereira's lab at UCSF. So we had made this model several years ago where we can turn autophagy on and off using a TET-inducible system where we overexpress the dominant negative ATG4B. This basically sequesters pro-LC3 and stops the formation of autophagosomes. We demonstrated that this inhibits autophagy and is reversible. And what was interesting about this is when you turn off autophagy by feeding the mice doxycycline, you significantly impair the growth of fully formed pancreas cancer. So this was done after the tumors had already formed. And this was the first time, at least in our hands, that we were actually seeing frank regressions in these tumors. As I mentioned, one of the striking things that we noticed was that in the tumors where autophagy was inhibited, there was a substantial increase in inflammatory cells. You can even see it on the H&Es. And we started exploring that. We focused initially on the increased infiltration of macrophages into these tumors, and showed that the macrophages were actually important for the anti-tumor effect. But we wanted to explore this further and look at other aspects of the immune system. In particular, because pancreas cancer is the poster child of resistance to checkpoint blockade. If you look at all the early trials, which were all comers, pancreas cancers were the ones that didn't have any responses at all. It's known as the canonical cold tumor. MHC1, which I don't have to explain to most people in this audience, is a critical component of T cell recognition and killing of tumor cells. And this has been seen to be frequently downregulated or lost in multiple cancers, including pancreas cancer. And this is one of the mechanisms, one of probably several mechanisms implicated in resistance to immune checkpoint blockade. But unlike other types of cancer, we don't see any mutations in class 1 that would explain why it would be downregulated. So we decided to look at this further. We took organoids that we made from normal pancreata versus tumor organoids. And similar to what had been published before, you could see that on the cell surface, there was significantly less class 1 expression. When you just looked across a panel of human cell line, pancreatic cancer cell lines and compared that to normal ductal epithelial cells, while many had lower levels of total MHC1, it was pretty variable and heterogeneous. So this didn't really explain potentially why this was occurring. However, when you looked at the localization of class 1, this was very revealing. So again, this is a non-transformed human pancreatic ductal epithelial cell stained for endogenous MHC1. You can see the majority of the expression is not surprisingly on the cell surface. What was intriguing was when we looked across human pancreatic cancer cell lines, the pattern of expression was quite distinct. It was punctate. It co-localized really well with lysosomes using a lysosomal marker LAMP1. You can just see that quantified over here. I mentioned and showed that pancreas cancers have very high levels of autophagy. One of the ways which proteins do get to the lysosome is through autophagy. So we wanted to see if perhaps that was playing a role here. And we were able to show that class MHC1 in these punctate locations co-localizes extremely well with markers for autophagosomes, such as LC3B. So it appears that in pancreas cancers, that the majority of the LC3 of the MHC1 is localized in autophagosomes and lysosomes, not on the cell surface. When you inhibit autophagy, this is knocking down one of the canonical ATG genes, ATG3. You get an upregulation of MHC1, shown here by total, and then in various B and C as well. You can look at this by doing immunofluorescence, shown here. When you knock down ATG3 in 2 different pancreatic cancer cell lines, you get significantly increased expression of class 1. This class 1 is increased on the cell surface, shown here when you knock down ATG3 and another critical autophagy gene, ATG7, you get an increase in class 1 expression on the surface of 2 different pancreatic cancer cell lines. This is the mouse system that we use with the inducible dominant negative ATG4B that I showed you, and they grow these cells in organoids. When you put them on docks, you get a significant inhibition of autophagic flux, which tracks perfectly with a substantial increase in class 1 on the surface of these cells. Through a lot of work, we were able to identify a new selective autophagy pathway where the autophagy receptor NBR1 binds to and recruits MHC1 and allows it to bring it to the autophagosomes and then the lysosomes. This is a selective autophagy pathway. And just like when you inhibit total macroautophagy by knocking down things like ATG3, 5, and 7, when you knock down the autophagy receptor NBR1, you could significantly increase the cell surface expression of class 1 in human pancreatic cancer cell lines. So this was all in vitro. We wanted to move in vivo, so we took advantage of our syngeneic ATG4B dominant negative system. Mice were, the cells were implanted into the pancreas of mice. They were put on doxycycline. And then, as I showed you before in the genetically engineered mouse model, when you inhibit autophagy by using this dominant negative, you significantly decrease tumor burden in the pancreas. Now, when you look at these tumors, as I mentioned before, this is the control tumors. These are the tumors where autophagy is inhibited. You can see even on H&E that there's a substantial inflammatory infiltration into these tumors. And when we characterize this more extensively, well, first we actually verified that MHC1 went up on the surface of these tumors where autophagy was inhibited, and this was done by flow cytometry. And when we did some immunophenotyping, one of the most striking phenotypes that we saw was a significant increase infiltration of CD8 positive T cells. This is shown by FACS, and this is shown by immunofluorescence. And we've repeated all these experiments in multiple different independently derived KPC lines. So this is in primary tumors. We wanted to see if the same thing was true in metastasis. So we utilized a liver metastasis model, which is basically performed by injecting the cells into the spleen. You can see you get very nice liver metastasis. When you inhibit autophagy in these cells, you can see that they are condensed and shrunk and quite small, the liver metastasis. You can quantify this by weighing the livers, and you can see there's a substantial reduction in metastatic burden in the liver from pancreatic cancer cells when you inhibit autophagy. Again, These liver metastases where autophagy is inhibited have significantly more class 1 on the surface. And again, we saw a significant infiltration of CD8 positive T cells. And this is just showing the same thing by immunofluorescence. So mechanistically, we wanted to understand the role of the CD8 T cells functionally in this process. So again, when you inhibit autophagy, you get a decrease in tumor growth in this model. When you deplete CD8 positive T cells in the control tumors, you really don't get any effect. This again is a nice indicator of what's seen in human patients, that T cells really don't have much of a role in the anti-tumor response of these tumors. However, in the autophagy inhibited tumors, you get a significant rescue of the tumor inhibition when you deplete CD8 positive T cells. Similarly, you can perform the same experiments in BATF3 knockout mice. These have dysfunctional CD103 positive dendritic cells. These are important for effective cross-presentation. It's been shown to be important in several tumor models for CD8 infiltration into the tumors. Again, in control tumors, really not much of an effect. When you inhibit autophagy in wild-type mice, again, a decrease in tumor growth, and a large fraction of this is lost when there's no CD103 positive functional dendritic cells. Lastly, we wanted to functionally link the increased class 1 on the cell surface to this. So we we used a hairpin to beta-2 microglobulin, which is a key component of the class 1 complex. And again, in control tumors, there's really not much of an effect, but when you knock down B2M in autophagy inhibited tumors, you can get a significant rescue of the tumor growth, indicating the importance of the increased cell surface class 1 in this phenotype. So next, the logical question was, would this synergize with the immune checkpoint blockade? So we did an experiment where we used PD-1 blocking antibodies and our autophagy dominant negative model, and we saw really no effect. So we did it again, except this time combined the anti-PD-1 with anti-CTLA-4. So again, in autophagy inhibited cells, you get a decrease in tumor growth. Another important control, again, showing dual immune checkpoint blockade really has no effect on control tumors, similar to human patients. However, when you add dual immune checkpoint blockade to the ATG4B dominant negative, you get a further increase in tumor shrinkage and a significant synergistic increase in CD8 positive T cells. This is by FACS, and this is showing by immunofluorescence. So I've mentioned chloroquine and hydroxychloroquine a number of times in this talk. These are, while not potent or selective autophagy inhibitors, are relatively safe medications and are being tested in the clinic as autophagy inhibitors. So from a translational standpoint, we wanted to see if we could get a response by combining chloroquine with dual checkpoint blockade. So first, we used a reporter system in these cells to actually show at least we were getting some level of inhibition of autophagic flux in these tumors, shown here by this GFP-RFP reporter. I'm not going to go into how it works, but it's certainly not a complete inhibition of autophagic flux, but you are getting a significant decrease in autophagic flux. Given the autophagy inhibition, we saw not too much effect of chloroquine on its own. However, when you combined it with immune checkpoint blockade, you got a synergistic anti-tumor response and saw a significant increase in infiltrating CD8 positive T cells. So the model that we've come up with is that pancreatic cancer cells, because of their high autophagic flux, have relatively low levels of cell surface levels of MHC class 1 on the surface through this selective autophagy pathway that we've identified. If you inhibit the lysosome or you inhibit autophagy, you decrease the recycling or the targeting of MHC1 to autophagosomes and lysosomes and increase the expression of the cell surface. This allows CD8 positive T cells to recognize these tumor cells and kill them. So in conclusion, elevated autophagy appears to be a major mechanism of immune evasion in pancreas cancer, and maybe one of the explanations for the lack of response to immune checkpoint blockade. autophagy inhibition in pancreas cancers decreases tumor growth, increases class 1 expression on the surface and CD8 T cell infiltration. We see synergistic responses with autophagy inhibition and immune checkpoint blockade. And this data suggests that at least part of the anti-tumor response of autophagy inhibition is immune mediated. And generally, conclusions for our work in autophagy is that there are multiple mechanisms by which autophagy supports pancreas cancer growth through cell autonomous and non-cell autonomous mechanisms. These include support of mitochondrial metabolism, maintenance of nutrient crosstalks with the stellate cells. It provides resistance to cancer therapies. And what I just showed you is that it may promote evasion of the immune system. And we're particularly excited about some interesting combinations that Channing Durr's lab and Marty McMahon's lab have shown, where you see some nice synergy with hydroxychloroquine and MEK and ERK inhibitors. And now multiple clinical trials are open to test this, including at Dana-Farber. But what, for us, what's been most exciting is now we're starting to see signs of efficacy of this approach in the clinic. There were 2 randomized clinical trials published last year. These were both randomized phase 2s. The first one was in a neoadjuvant approach where all patients got gemabraxane, the common doublet that's used in this disease, and they're randomized to get hydroxychloroquine or not. And all patients went on to surgical resection, and then they looked at the responses in histological specimens. The patients who were randomized to hydroxychloroquine had substantially increased tumor responses, more apoptosis in the tumors. Their tumor marker CA19-9 went down when they got hydroxychloroquine, and there was less involved lymph nodes. And what was very exciting to us, and this was just presented at ASCO, and we think provides some confirmation to what we saw in our mouse systems, is that the patients who were randomized to the hydroxychloroquine arm had significantly more T cell, particularly CD8 T cell infiltration in these tumors. And then a second clinical trial, which is really the same backbone. This was done by Stand Up to Cancer, except the top one was in the localized setting. This is in the metastatic setting. While survival was not improved in this trial, there's a substantially increased response rate. So we think these 2 trials really indicate there is activity in this disease, and that we're hoping with some of the new trials in development, either with MEK or ERK inhibition or now with immune checkpoint blockade, that we may start to see even more activity and more responses. So I'd like to thank all the people in my lab, past and present, who did the work. I think I thanked pretty much everyone during the particular parts of the talk. I think the only one I might not have mentioned is Kasuki Yamamoto, who from my lab spearheaded the MHC1 studies. Again, many collaborations, and I'll just mention, Joe Mancius, who now has my lab at the Dana-Farber and is doing very well as an independent investigator, remains a fantastic collaborator to this day, and Andy Aguirre, who is continues to be a fantastic collaborator. So I could stop here and take any questions.
All right, so we have several questions. I'm going to try to get to all of them. So Alec, keep your answers short.
I have answers. Do my best.
Why do you think serine glycine deprivation induces stalling at only some serine codons and not others?
It's a fantastic question. Robert Bond, who is the postdoc who did that work, this is going to be the focus of his lab. We have some ideas in that it may have to do with the amount of a particular cognate tRNA that's present, but we really don't have the definitive answer. It's an incredibly interesting question that I think holds important promise, not only for cancer, but for other metabolic diseases. And Robert's really intently focused on this.
So on the same theme, is the difference between serine dependent and independent tumors genetic, or what is the mechanism?
So it brings up a couple of important questions. One is, and someone else may have asked this, but why would a tumor have any advantage of losing serine biosynthesis? And we still don't really know the answer to that. We're trying to understand why. I mean, one of the potentials is that perhaps there's a toxic intermediate produced when you're making serine. If you don't need to, it's energy consuming, but we don't really understand why. It doesn't appear to be genetic. There doesn't appear to be mutations or genetic deletions in this. We think it's probably epigenetic. When you look, there's differences in some of the epigenetic marks in the promoters of PHGDH and PSAT in the different serine dependent versus independent cell lines. So we don't exactly know. It's not genetic, though. It does not appear to be genetic.
Okay. So this is coming to the sort of the latter part of your talk. Is MHC1 necessarily being degraded by autophagy or just recruited to the autophagosome? And how specific is NBR1 in recruiting MHC1 versus other surface proteins?
So it is causing the degradation. So you can increase total levels when you block the lysosome. So it is targeting it to the autophagosome and then trafficking it to the lysosome where it's degraded. MBR1 has other targets, but it appears to be a fairly specific interaction with MHC1 in that it appears to be ubiquitin dependent. So MHC1 gets monoubiquitinated, and then MBR1 through its ubiquitin binding domain binds to MHC1. But whether or not other cell surface proteins are also degraded by autophagy via NBR1 or other adapters, we don't know.
Why are PDACs not more sensitive to NK cell killing if they have downregulation of MHCs on the surface? I thought this was an interesting question.
Yeah, no, this is an important question. Obviously, I am the furthest thing from immunologist. I got dragged kicking and screaming into having to sort of study this. What we think is happening is that it's not, class 1 is not gone. It's not, deleted. It's just basally lower through this recycling of class 1. So it's probably enough there to not have them killed by NK cells, but not enough really to have a robust T cell response. That's the best answer that we've been able to come up with.
All right, so the last one on my list, this is from Stephanie Dugan. I think she asks, Alec, you mentioned briefly that you had looked at macrophage depletion in the autophagy inhibition mechanism of action and potentially seen a role for macrophages. Can you elaborate on that?
Yeah, so hi Stephanie. So in the original paper where we published that model with the ATG4B dominant negative, we focused a lot on the macrophages. And what we did was that we sort of used what you would consider a very blunt tool. We used the liposomal claudronate to deplete the macrophages. And we saw that we lost some of the anti-tumor effect. We're actually going back now and trying to do this in a more targeted way because, as you know better than I, the liposomal claudronate will affect sort of all phagocytic cells, potentially dendritic cells as well. So we're now going back and trying to repeat those experiments to try and understand, specifically macrophages, what type of macrophages are dendritic cells, how are dendritic cells evolved, and how do the macrophages and T cells talk to each other?
So just, I feel I have to ask one question. It's more on the clinical side, but I know when you started, the trials with the chloroquine or the other autophagy inhibitors, has the combination of immunotherapy and that been done, or is it being done?
They're being developed now.
Okay, because it says, show that it synergizes, but I'm wondering whether it's come to the clinic yet.
Those trials are under development. The trials that are now in clinic are combining hydroxychloroquine with MEK and ERK inhibitors, because there's some nice synergy in preclinical models there. One of the issues has been, and I alluded to this, is that hydroxychloroquine is not a great autophagy inhibitor. It is, well tolerated and patients can take it. They could take relatively high doses of it, but it doesn't it doesn't completely block autophagy and probably doesn't block the majority of it. So I think really to move the field forward, we and others are hopeful that there will be sort of more potent and selective autophagy inhibitors.
So a question just came in from Rakesh Jain. Congratulations, Alex, on this elegant work. This is, I guess, a comment. Increase in MHC1 explains cancer cells by CD8 T cells. How does chloroquine increase their delivery to PDACs?
Rakesh, it is a great question. One of the things that this model is contingent on is that there has to be the presence of at least some CD8 positive T cells in the tumor in order for this to work. Then we believe the signal gets amplified by dendritic cells, et cetera. But your point about does it normalize vessels, that's been shown, as you're pointing out.
No, I think that's what Dr. Jain is saying. Chloroquine and hydrochloroquine have been shown to normalize tumor vessels, and vascular normalization can improve the delivery of the CD8 positive cells in tumors. So this mechanism of more CD8 cells with chloroquine and PDAC.
It's possible that it contributes to it. There is one of the things that's really interesting, and you know this, Rakesh, is that there's incredible heterogeneity in the amount of T cells, particularly in patients. The mouse model that we use is in a very sort of extreme example of a very poor T cell depleted environment. Whether or not in patients, I think there's a couple of things that this brings up. One is that this therapy will work better or likely to work better in patients where there's at least some CD8 positive T cells initially in the tumor. Those that have virtually none, this may not work. It also brings up the fact that the patients who have less class 1 on their surface, because they have higher levels of autophagy, may also be more prone for this therapy to work. But I think you're right. The concept of vascular normalization, it very well could contribute to this. I mean, even in the human clinical trials where they put hydroxychloroquine without checkpoint blockade, they did see more CD8 positive T cells. So there may be a component of that as well.
All right. If there are no more questions, I will thank Alec again for coming or being here on Zoom and telling us about his fantastic work. Thank you, Alec.
Thanks, Dependencies. Thank you, everybody.